Hi there guys, and welcome to another video of A-Level Biology. This is Phillips. This is about photosynthesis, and today we're going to be looking at the light-dependent stage of photosynthesis. So quite a tricky one. You probably will need to support this by reading the books and looking for sources elsewhere, but we'll try and go through it and make it as easy to understand as possible. So, just to remind you, if you haven't watched the last video, photosynthesis happens in somewhere called in the membranes of the thylakoids, called the lamellae, and particularly focuses on somewhere called the primary pigment reaction center. And basically, all the different pigments of light in plants in the thylakoids focus all the energy onto this primary pigment center, and we're about to see what happens at that with that energy in a second. So we have pigments arranged in light harvesting clusters known as photosystems. Within a photosystem, there's several hundred accessory pigments which capture light from all different wavelengths, and they all focus it on one primary pigment, which is normally chlorophyll A. So the light-dependent reaction, then, it's important to get an overall view of it. So the light-dependent reaction, the inputs of the light-dependent reaction are light, which obviously we're not going to be measuring, but we'll be saying a photon of light, water, which is obviously a key photosynthetic ingredient, and something called oxidized NADP. So at the moment, we're not going to be talking about carbon dioxide because that's in the second stage of reactions, which we'll talk about in the next video. The products we're going to be looking at are ATP, which you might think, well, it's not respiration. Why is that a product? Okay. Oxygen, which is just diffused straight out of the plant, and reduced NADP. So anyone that's watched my videos on respiration will know we've talked about reduced NAD carrying hydrogen to the final stage of respiration. This is the same here. Reduced NADP carries hydrogen to the final stage of photosynthesis, as does ATP has to go to the final stage of photosynthesis to be used up there. So reduced NADP is a coenzyme again, which carries hydrogen. I just think of it as the NAD for photosynthesis, NADP. Okay, so let's go through it. So what you need to know basically is that there's two essential reactions: the light-dependent reaction, the one that needs light, the light reaction is often called, we're going to talk about today. That splits into two bits: non-cyclic photophosphorylation and cyclic photophosphorylation. And then there is the light-independent reaction, or the dark stage, which we'll talk about next time, so you can link these together. So there are basically two pictures. If you learn these pictures and you can talk your way through these two pictures, you'll get decent marks in your test on this. So basically, this shows you the very similar diagram if you've done photosynthesis, and we're doing now obviously, but if you've done the respiration module, very similar looking thing almost to the inner mitochondrial membrane, which is used in the oxidative phosphorylation reaction. So this is now where we're going to be doing this reaction, and it's got a similar purpose: generating ATP in order to power the later stages of respiration. So let's have a look at this and just see what we can come up with together. So if we start off then, this reaction, we're talking about light being absorbed. So you can see here and here, we've got photosystem 1, photosystem 2. Photosystem 1, photosystem 2 are part of the lamella of the membranes. Slightly different molecules of chlorophyll A. One of them absorbing chlorophyll A at light at 700 nanometers wavelength, one of them 680. But irrelevant for your understanding at the minute, they absorb light. What they do with that light then is they funnel it down to the main reaction, as said, and what that energy does is it excites an electron. So the whole point of absorbing light is to excite an electron to give it energy. Now, we'll come back to water in a second. Let's just look at what happens to that electron. That electron becomes highly energized, and it is then passed down a series of electron carriers, which you can see here in the orange, to the next photosystem. By passing those electrons through, what that does is it allows hydrogen to be passed from the stroma into the thylakoid space. So very similar to respiration, moving hydrogen molecules through. So you're creating a hydrogen concentration here, okay? And those hydrogens then are passed through an ATP synthase, which generates ATP, okay? So slight error on this picture, that should be a molecule of ADP there. So we generate some ATP by moving by motive force, by causing the movement of the ATP synthase from the hydrogens moving through. They go to the final stage in respiration. The hydrogens then are mopped up by the coenzyme NADP, which is oxidized at that point, picks up the hydrogens, become reduced NADP. Now, what else happens then? So once that electron gets to photosystem 1, that electron replaces an electron that was originally in photosystem 1 because the light energies come into photosystem 1 also. That electron has got excited, and that electron has gone through some electron carriers, the last one called ferredoxin, and that also attaches the NADP, helping to reduce it. So let's have a look then. What are we missing? Well, we've said about electrons. So the light energy goes in, it excites electrons, causes hydrogen movement. That then causes ATP synthase, the enzyme to turn, and that causes the ATP generation. The hydrogen and the electrons then are accepted by NADP, and that causes reduced NADP. But we've still got this molecule of water here. So why is water essential? Well, if you imagine if water, what happens in water is light energy hits water and it causes it to split into two molecules. It causes it to split into hydrogen ions, positive ions, and oxygen. But when they split, they also release two electrons. So what those electrons do is they go in to replace the electron that has been excited and has left photosystem 1, thus keeping the system going. Okay, oxygen just diffuses straight out of the cell. Okay, and the hydrogens can also add to this photochemical hydrogen gradient to leave via ATP synthase. So let's put it into words. So first thing that happens is photoionization. So that is where the energy comes in, it excites an electron in photosystem 1, it excites an electron in photosystem 2, and those electrons leave the photosystems and go to the electron carriers. Okay, so that's your first stage. Each photon of light excites two electrons, so each time we're talking about two electrons from each carrier, from the chlorophyll A, which is holding on to it. Electrons captured then and passed on the electron carrier chain, releasing energy. That causes proton gradient because it uses it to pump the hydrogens into this area here, and that builds up there in the thylakoid space. The hydrogens then move through the ATP synthase, generating ATP, hence the name photophosphorylation. And ATP sends light to the independent stage, to the dark, sends energy, ATP goes to the light stage. So what didn't we mention there? We just didn't mention the water. So if you're going to talk about it in stages, this is how I would do it. So I'd say number 1, the photosystem absorbs the light that excites two electrons from the magnesium in photosystem 1 and 2, and the electrons move into the electron carriers at number 2. And number 2, the electron carriers, as the electrons move through them, they move hydrogens through into the intermembrane space, and that builds up your hydrogen gradient. To replace the electrons lost at number 1, water is split in a reaction called photolysis, and the water molecule electrons then go in to replace the electrons lost from the magnesium in chlorophyll A at photosystem 2. Then we have the electrons getting passed through the electron carrier chain, and if they get to photosystem 1, they replace the electrons from photosystem 1, and again they then get excited again and pass through the last few electron carriers. The buildup of hydrogen in the intermembrane space then builds up, and the buildup of hydrogen then moves via what we call proton motive force through the ATP synthase. That generates ATP, which goes to the dark stage, or the Calvin cycle as it's called. So I've drawn that by showing it here on this slide. The hydrogens and the electrons that have been moving through either the electron carriers or the ATP synthase react with NADP, or oxidized NADP, to make reduced NADP, and then those molecules also enter the Calvin cycle, which we'll look at in the next video. Now, there are two types of reactions actually going on there. One of them that we've already studied a lot, which is what we've just done through, is non-cyclic photophosphorylation. So if you imagine the picture we just looked at, the two photosystems are called photosystem 1 and photosystem 2. They're discovered, their names are basically the order they were discovered. So the second one is actually photosystem 1, and the first one that we talk about is photosystem 2. So we start with photosystem 2. We've just got the same picture here. We've got water splits into two, hydrogen and oxygen, which also releases 2 electrons. Those 2 electrons replace the electrons excited by water and excited by the light in photoionization, and they go up through the electron carriers, increasing their energy levels. As they decrease their energy levels, they pump hydrogens through the molecule, and then they replace the electrons at photosystem 1, and the same happens again. They go through a series of electron carriers until they combine with NADP using the enzyme NADP reductase to make reduced NADP. And that is non-cyclic photophosphorylation. However, there is another version called cyclic photophosphorylation, and this is in the photosystem 1, which don't forget is a second photosystem. Some of the electrons excited during photoionization do not fully escape the system. So they get excited, but they go through a series of electron carriers and then back to photosystem 1. So in terms of cyclic phosphorylation, it's the light absorbed by the electrons in photosystem 1, excites it, gives it energy, makes it leave the photosystem, passes down a series of electron carriers before returning back to the chlorophyll molecule into the Mg. Now, why do they think this happens? They think this is a different form of ATP generation, which is just used to open and close the guard cells for stomata. And therefore, non-cyclic photophosphorylation is what we've talked through in much more detail. Starts in photosystem 1, electrons get excited, so in photosystem 2, then through photosystem 1, and then through a couple of electron carriers and reduced NADP at the end. So let's have a look to see if we could do this table. Cyclic and non-cyclic photophosphorylation. We look at non-cyclic first, which is the first one we went through in a lot more detail. It does involve photosystem 1. It does involve photosystem 2. Where does photosystem 1 obtain its replacement electrons from? Well, it gets its electrons from the ones that are lost from photosystem 2. Okay, and where does photosystem 2 get its electrons from? It gets them from the water molecules that split up and use the electrons as and when they're needed. Now, is ATP made? Yes, ATP is made. And is reduced NADP made? Yes, it is. When we look at cyclic photophosphorylation, is photosystem 1 involved? Yes, it is. But it happens in a cyclic manner, so you don't involve photosystem 2. Where does photosystem 1 obtain its replacement electrons from? Well, it's cyclic, so it obtains its own replacement electrons from itself. Photosystem 2 isn't involved, so that question is not applicable. And then ATP is ATP made. ATP is made to be used in the chlorophyll to open and in the stomata to open and close the guard cells. And is reduced NADP made? No, it isn't, because it's not involved in the hydrogen pumping across the membrane. So that's been useful for you guys, and I know it's quite a tricky subject, so obviously have a good look at the books on that when you can. But for the moment, we'll leave it there. Thank you very much. See you soon.